All right, good afternoon. My name is Christopher Higgins, and I'm giving the presentation on applications of whole slide imaging into neuroscience. What we've seen over the past bunch of years is that whole slide imaging, which is usually used for digital pathology for clinical use, is now being used in neuroscience for the ability to look at high resolution images in a field of view, but to be able to zoom out to see what parts of the brain that the neurons are connected to be able to see in different areas of the brain where the signal or the neurons are connected to, and that's what digital pathology allows you to do. So today, researchers we've seen a shift, like I mentioned, of imaging just cells, individual patch clamping in tissue, and then acquiring a 3D montage of a set of magnifications. The problem, though, is that the current method in most labs right now is it's very labor intensive, and it can be very subjective when you're doing the stitching manually. And the idea is the researchers are looking to gain answers in a very short period of time. And the researchers are now adopting digital pathology to be able to capture the image and then do a whole slide analysis on the images itself. So the different types of scanning is available right now. There is the area scan method and the line scan method. And the difference between those two is that a line scanner continuously moves over the tissue, so the slide is moving in one direction and is able to capture it kind of like a fax machine. And but the disadvantage of those systems usually is resolution. They're very fast, but not really accurate in the resolution. And then the traditional area scan is very good, but you would have to start and stop the stage. But technology has gotten very good with the stages, where you're able to have precise XY movements, and then you have the ability to have high resolution pictures. And so the area scan methods allow you to have many different magnifications that you're able to capture with individual objectives. The idea of capturing a whole entire glass slide for digital pathology allows you to revisit the slide offline on a computer and be able to zoom in and out of the system, out of the actual image itself, with the resolution of the actual objective you captured at. And so if you would like to look at it at 40x, you capture the whole entire slide at 40x, and it stitches very nicely, and you usually don't see any seams or overlap. But then there's a 1D or one plane is usually kind of limited, when you have a very thick sample. And so the idea, the next step is to be able to capture multiple Z levels so you can focus or simulate the microscope, or to combine the images to be able to compress into one perfect plane. As a neuron, it'll dip in and out of a section. The usual typical sections are about 50 microns thick. And the idea is we would like to see them all in the same focal plane so we can map it out. So multiple Z acquisition and then being able to post-process that into some one plane is available today with most systems. So today, what we look at is in mouse brains, the connectivity was one of the first projects that were out there a few years ago, where we would rapidly try to capture as many brains or sections as possible and being able to connect a neural network for the mouse brain itself. And then we also see that a lot of groups are also looking at where creating an atlas for gene expression throughout the adult mouse brain and even some younger mouse brains. And then later on, we'll look at the idea of 3D neural visualization. So in this example image here, this was captured at 20x. This is a 2x3 slide, and you can see from the call out to the side right there, you can see the high resolution zoom in on the image, where you can see the astrocytes. The scan area is 44.9 millimeters by 54.4 millimeters, and a size like that is a 57 gigabyte image. It's quite large, but these scanners can capture these slides within 10 or 20 minutes for that size. A typical 15 by 15 usually could take really just several minutes to capture per region of interest. So how many pixels is this image? Well, it's about 20 billion pixels. We're looking at 129,000 by 157,000 pixels. So it's the ability to have macro to micro imaging in the same image itself. So you're able to deliver the resolution that you're doing your research at. And then the ability was added on a few years later for the idea of fluorescence imaging and montaging of fluorescence. And you can do, in this image here, we have an overview that was done in GFP, and then the detailed scans, the regions of interest of the multicolor brains were all done at 20x at 3 channels. And it could be zoomed in like this. And so this is a region of interest at 20x. But as most of the mouse connectome project of mapping is finishing out, or maybe 3/4 of the way through, we're noticing that there's a shift to moving up to other size brains, and for neuroscience, and human brain has been coming up a lot lately. The idea is to create an atlas for the human brain development, and then we've seen a lot of shift also to traumatic brain injury from radiology to micro, and being able to correlate the difference between the micro and the radiology. So building a 3D model in brain injury for DP and comparing it with radiology is now being the shift of adding extra modalities. This is an example of a 4x5 slide with a human brain slice, and this also was a very large, it's about 100 gigabyte image uncompressed, but we have pixel resolution of 0.32 microns per pixel when you zoom into the image itself. This is a image of a region of interest for a brain slice that was infected with MS. And you can see the individual fluorescent channels that were captured beforehand and then combined in pseudo color for red and green. As we look at the whole entire slice here, actually this was captured in 3D, so it's a little soft because if you focus and zoom up and down through the sample itself, you're able to see the individual fine filaments of the brain sample there. And then if we zoom in into that, you can see this very nice image. And so you have that resolution. So this was actually captured with a 30x silicone media objective, especially the objective that we make that they're able to see deep and also match the refractive index of the sample itself. But as we talk about mouse mapping, brain mapping, the USC has been part of the Connectome project for quite a while and NIH funding, and they are imaging up to about 1,000 brains right now, and they've posted over 200 brains onto their website. And this will help researchers get a better understanding of how different the brain structure is and organized with their atlas. So their atlas, this is a snapshot from their website where they're looking at the animal one, and they have that as a Nissl stain, and they're able to see that in bright field. And then their app will allow you to turn on the fluorescent channels of the areas of interest that you're looking for, and being able to check out each individual serial section and where you might be within the brain. This here is another example of their map viewer navigator, and looking at the different sections at different levels within the brain. This website gets 20,000 unique visitors per year, so this helps neuroscientists to look at this atlas and compare it to their own research. But sometimes looking back and forth between a website and also a printed book on the atlas can get very tedious, and if we can create an application that can utilize, like Allen Institute, at open source for their brain atlas, then Visiopharm has taken advantage of that, and they're able to take that Allen Institute Brain Atlas, and then they're able to automatically detect regions of interest. And I think a better way would be the picture here. So you have the original brain slice that you have captured, and then you overlay the atlas over the brain slice, and then from there it'll align it to the atlas, and then you're able to pick out the section over the region of interest that you're looking to do your research. And this is easily done within their software. From there, you can see in this picture here on the simplified workflow that the user has picked cerebellum, and then they can click on each different area, brainstem, cerebellum, or cerebrum, and they're able to check those off, and then they would be highlighted effectively on the atlas itself. But from there, after they do their research within those different areas, and they're looking to start their measurements and accounting in the different areas, a lot of them will like to also do some type of visualization. And so there's a lot of commercially available visualization products out there, and I'm just going to give you some examples. So this is a typical mouse brain slide with 12 sections on it, and they're captured 3-channel fluorescence. And in this user group here, usually a mouse section up to 140 sections on about 10 slides. And what we'll do with that is we'll use a software package to align all the brain sections or the regions of interest into a volume. And then after they're all aligned, then you will push it out to a visualization. So then the user can now look at where the activity is localized within the brain and be able to do some analysis from that. But a lot of times when you're looking at the global view of an area, the cells are too small to count visually. So then with the software, you're allowed to be able to zoom in to an area and then readjust the visualization for that area itself. And from there, when you're looking at those green cell bodies, then you can push that out to an image analysis for a 3D volume analysis. Another example here is another package from Biplane, the Amera software, where we capture also 140 sections. And in this section here, this is a brain that was infected by a rabies virus, and they're looking at the size of the neurons. And they have captured this in multiple sections and extended focal imaging, and they were able to push this out to an image visualization like this. So with this visualization, you're able to verify where the activity is happening from the virus injection, and if the virus has actually moved from different parts of the brain or did it stay in the same area. And also we can see with the color coding later on in this visualization, the length of the neurons. So digital pathology for neuroscience is actually becoming a mainstream product where you're going to go from micro to macro, and the ability to capture many slides and being able to process through the research a lot faster. A lot of these systems are very automated within bright field and fluorescence, and then you would concentrate on the research itself and the data and analytics that are available today. And then you'll be able to also, because it's digitized, there will be fluorescence won't photo bleach or dissolve over time. And then if you tag your images effectively, you can data mine it and you can do image comparisons, side by side areas. You can have serial sections that can be combined as easily with some visualization very easily. Thank you.